Hi, this is Dr. Jabali, and in this video here, we're going to work on metabolism, which is topic 8.1, which is everything related to the chemical reactions that happen in your body, focusing more on the enzyme reactions. So, what are the things that we're going to look at topic 8.1? There's different types of metabolic pathways, all of them based on enzyme. We're going to talk about enzyme knowing the activation energy. We're going to talk about different types of enzyme inhibitors, such as the competitive and non-competitive. We're going to talk about the end product inhibition and how it controls the enzyme actions. And then we're going to see the one example, which is the end product inhibition of threonine turning into isoleucine. We're also going to look at the database to identify potential new anti-malarial drugs, distinguish the different types of inhibition graphs, which is very much common seen in IB exams, and calculating and plotting range of reactions of raw experimental results. So with that in mind, let's start with the big thing. What is a metabolism? That can be also common definitions, and you can use this in the big essays, which is basically the sum of all chemical reactions that occur in you as a living organism. So think of every single thing that is happening in your body, all the chemicals that is going, and that's metabolism. So not only are you making things, but you're breaking things inside of your body. So that's why we have these two words here. We have anabolism and catabolism. So anabolism, it's actually using energy to build big stuff from the small stuff. And catabolism is using energy to break down stuff from basically the big molecules into the small ones. So here is a very good chart that you should know it really well because it can come as a short answer question. So first anabolic, it's building, while catabolic is breaking. The anabolic is endergonic, which in this case, that means you're using the energy. And exergonic, you can see X as in this means going out, so energy is released. And you can think of N as in. And then anabolic is biosynthetic, which that means you're building something on. As you can see, the word synthetic means like synthesis of making. And degradative means degradation, which that means you're breaking it down. An example of anabolic is photosynthesis because you're making glucose. While we have here cellular respiration, which is the breaking of glucose. So this is a perfect chart if they ever ask you to explain the differences between catabolic and anabolic. This chart, if you know it really well, you get all the points. So now that we're talking about metabolic pathways, it's a chemical reaction. Sometimes we can make those chemical reactions go faster. How is done that? Well, it's done with what we call the enzymes. Why? Because they're catalysts, which means they speed up the reaction. Now, any reaction that happens in a specific sequence, we call it a biochemical pathway. Because again, bio here, you're just saying it's life. And chemical, that means we're using chemicals such as chemistry here in order for things to work in life. So we usually have a substrate A plus a substrate B making a product C. And right here, usually we'll have the enzyme that's making this function go forward faster. Now, in our genetic makeup, we also can make a specific type of enzyme that will work in a specific type of reaction. And here is just a very small sample of different types of chemical reactions with enzymes located in our body. So, since we're talking about enzymes, let's go in deeper. And this is more about 2.5, so it's kind of review for information. So first, we have to know that enzymes are actually made of proteins, and their shape is very important for their functions. Especially why? Because of the active site right here. The shape of the active site makes a big deal. The shape of the active site will tell with which side the substrate goes. So that means the shape of the active site should be very much closely related to the shape of the substrate. Now, what is the point of these enzymes? Usually they're used to break things down or to make big molecules in your body. And it's known as a catalyst, which means speed up the rate of the chemical reaction. Now, they might ask you, how does the enzyme speed up the reaction? This is the moment you have to mention about the activation energy, which is the amount of energy needed to make the reaction go forward. So the enzyme, what it does is lower the activation energy. Another thing that you need to know is that most of the enzymes end with A's at the end of the name. So for example, sucrase, which breaks down sucrose, maltase, which break down maltose. So you can easily see the A's at the end. The A's, if you see that word, those three little letters, that means we're talking about an enzyme. So moving on, we have now cofactors. What are cofactors? These are just bound on the sides of an enzyme, and they usually help. So examples of cofactors can be vitamins. So you can have certain vitamins bound to the enzyme, or even minerals. They just bind and make the enzyme function more. Now, these cofactors are usually not protein because we know the enzymes are made of protein. So they're usually also known as prosthetic groups. So when I say prosthetic groups, I am talking about things that are not protein, such as vitamins and minerals. Like here you have nitrogenase enzyme, which binds with iron. And iron, for example, here is not a protein, it's just a mineral, or should I say an element in the periodic table. Now, the mechanism of enzyme action, you should know this already from 2.5. But to continue here, we have an enzyme, as you can see here. And enzyme always has an active site, these two little entrants on the enzyme. Now, these active sites will have to allow the substrate right here to join in. So when they attach, they become this thing called enzyme-substrate complex, which this gets you points by just writing enzyme-substrate complex. And then the enzyme speed up the reaction by lowering the activation energy, and you form the product right here, and you release the product out. So this is the process or the mechanism of enzyme action. It starts with the substrates binding to the enzyme through the active site, and then you form the enzyme-substrate complex, and then you produce a product and you release the product. That's the steps, or should I say the mechanism of enzyme action. So if they ever ask in the question, explain the mechanism of enzyme action, the first thing you mention everything about enzymes, them being a protein, they being having an active site, being specific to a substrate, then binding to the substrate, forming an enzyme substrate complex, and then speed up the reaction by lowering the activation energy, and eventually form a product and release. If you mention this, then you got the entire mechanism set. So let's talk about the law and key hypothesis. This is also 2.5. Remember, I'm doing a quick review on it. It just means that the enzyme has to be exact to the substrate. So that means you have a key and a lock, and they have to match. If they match, then they works. But if they don't match, they don't go in. It's the same thing with the substrate and the enzyme. If the substrate is exactly the shape of the active site of the enzyme, then it matches and works. Otherwise, it doesn't. So basically, the whole thing is saying is it has to be exact. However, for an HL course, there's something called induced fit hypothesis that they have come up. Now, they're saying that the active site doesn't have to be completely 100% exact as a substrate. They can be a little bit different. That means they have more chances of working with different substrates than just one. So what does that mean? That means, let's say they have a shape of a square. So you might have one that is like this. Now you're going to think that this cannot fit, but once you fit here, the active site kind of changes a bit just to be able to fit. So now you have fitted. So you're kind of changing a little bit of the active site to allow the substrate to go in. That's what a dose means. In dose means I'm kind of like forcing or influencing the active site to change a little bit. If that is so, that means you don't have to be one enzyme binding to one substrate. You can have that one enzyme changing its active, and now it has so many different 1, 2, 3, or 4 different types of substrates. Why? Because you can kind of manipulate or change the active site a bit, allowing for more options of substrates to attach. So what exactly is the whole point? And you need to know this. It's simply about changing the shape of the enzyme. Now, the word shape, we have a more specific name that we use in IB, which is known as conformation. So if you see the word conformation in the IB exams, it is the same thing as shape. So what it means, when the substrate combines with an enzyme, it induces a change in the enzyme's conformation. Now, where exactly is the point in which you're changing the enzyme? It is the active site. So it changes conformation in order to fit the substrate. Now, more specifically, what about the active site? What is inside the active site that is changing? It is the R groups of the amino acids. Because remember, enzymes is made of amino acids. So that means even the active site is made of amino acids and it's being changed. So that means that chain of amino acids is changing, and that means it changed the active site, which changed the enzyme. So by changing this, you make the chemical environment more suitable for who? For the substrate to attach. That way the substrate will attach inside, and this way lowering the activation energy. So here, let me just show you an example what I mean by that. So if you remember the enzyme here, let's put it enzyme, you have here which is known as the active site. Right? Now, if you think about the enzyme is basically made like a line here. Think of this whole line that I'm drawing here is actually a chain of protein. So this is what it looks like, right? More detail. Now this, imagine here, if I stretch this line, what is this line? It's made of a chain of amino acids connecting one after the other. So that means right here in the active site, this area, what do we have? We have a chain of amino acids. And these amino acids, what do they have? They have the R group extended, agree? Now, what happened is what if you change the R groups? What if I came and changed these R groups? So if you change these R groups, this was the beginning shape, but because I changed, let's say, one of the R groups, now it might look like this, the active site. You can see how it was completely changed from a square to become a little bit with a bump of circle. All of this because you changed the R group of who? Of the amino acids. So that's what it means by this whole process. So in detail, we do mention the word induce, right? That's forcing, that means who's causing the change? The substrate as it combines. But when the substrate touches the active site, induces a change. That change of the active site, more in detail, is the R group. So IB will ask you to expect you to know the most detailed part. You cannot just say, oh, the active site changed. So if you write here, the active site change, that's not enough. You got to be more in detail. It changed by what? By changing the R group of the amino acid. So all of this, who actually caused this whole thing? It all started with the substrate binding and inducing a change. So it is the substrate that induces the change. And where exactly? In the active site, more specifically the R group. If you understand this, then you're never going to get this point wrong. Next, let's talk about the chemical reaction and activation energy. We know the enzymes lower the activation energy, but you do need to know the graph. So what exactly is activation energy? It is the amount of energy you're putting into the reaction. So right here, we have in the y-axis the free energy. That means the energy that you use in the reaction. And in the x, we have the reaction happening. Reaction happening, we have the reactants and then the product. So it's the pathway which you're going from a reactant to make a product. So in this case, the reactants usually already have some kind of energy inside of them. But they need a little bit more energy here in order to make the transition from a reactant to become a product. Now, this energy right here that I just outlined, this energy that it needs to happen is called the activation energy. So once again, activation energy is the energy needed to proceed the reaction. So if they don't reach that energy, no reaction will happen. And when I say transition state is when you're going from a reactant to become a product. So what happens if you add an enzyme to this case? If you add an enzyme, you're actually going to reduce the amount. So you can see here on the graph that the amount has been reduced. The previous amount was this large right here, but because you add an enzyme, you don't need so much energy for the reaction to go forward, and that's what the enzyme do. Now imagine if you didn't have an enzyme. Of course, this is going to cause what? An increase in temperature. So why do you want the temperature to increase? Because you want the molecules to move faster, because if they move faster, more likely they will collide and cause the reaction. But the problem is, if you increase the temperature too much, you might destroy everything around you. So the enzymes comes like a savior, say, hey, I can make sure that the reaction go forward without having to increase the temperature. So it's kind of saving us from burning ourselves. So enzymes is a great thing to lower the activation energy, as you mentioned here. So this is a graph that usually IB exam will put it. They will put this line, and here they can say the reaction happening. And here they're going to put the free energy. And then they're going to show you a graph looking like this. And they're going to say that here is the reactant. And here is the product. And this is saying that is without an enzyme. And they will say, okay, if I do put enzyme, how things will change here. Obviously, you're going to have to make an enzyme that has a lower activation, because this here, this line that I just did red here, this is the amount that you need of energy, right? This chunk, that's the activation energy. But when you put an enzyme, what do you do with the energy? You reduce the energy. So that means the dotted line will come up here, and eventually it will line up with the rest to become a product. So here I'm showing that the energy has changed. A big energy here to a smaller energy. But this one is with an enzyme. So you should be able to draw this second line here. Now when drawing the second line, it's very important that you see that it's lower, right? It's lower than this one. And you got to say that they do eventually match in the line where it becomes a product. So it's only changing this area right here. It's like making a smaller mountain under the big mountain. Now let's talk about inhibitors. What are inhibitors? Those are the ones who basically reduce the rate of the enzyme reaction. That means they're slowing down the reaction. How do they do this? They usually will stop the enzyme. That's their purpose, is to block the enzyme. That's the main point of the inhibitors. And if they block the enzyme, what do? They reduce the reaction. Now, they usually do not destroy. They're just slowing them down. That's what they're doing. And they can usually work with just a little low concentration. So you don't need so much inhibitors. Just a little bit will cause the whole process to slow down. So there's different types of inhibitors, right? So it can be drugs and poisons. So sometimes that's why in medicine we give drugs for people to slow down some kind of infection, or poison can usually kill people because it's stopping most of our chemical reactions. So infections by virus can be treated with inhibitors to stop the viral enzyme protease, for instance. This means the virus cannot build a new protein coat and therefore cannot replicate. So for example, here penicillin is an example of an inhibitor. What it does do? It stops the bacterial enzyme that produces the protein to make the cell walls. So this is a perfect example of inhibitor. Penicillin breaking is stopping the production of the proteins that make the cell wall. So with that, let's look at two different types of inhibitors. We have a competitive one and we have a non-competitive one. These are two types of inhibitors that will try to stop the enzyme. However, the way they act is different. See, on the competitive one, and you can say the word competitive here as a compete, that means the inhibitor usually will have the same shape as the substrate. And they both will attack the same area, which is the active site. While the non-competitive, that means it doesn't even care if you have substrates. You can have a large amount of substrates and it makes no difference because they come from behind the enzyme, which is there's a place called the allosteric spot. So if they bind here, it would block any substrate. So you can have 1 million substrate, it doesn't matter. That inhibitor will stop all the enzymes from binding. So let's focus on each one more closely. When it comes to competitive here, they usually are reversible reaction. That means they can stop the enzyme for a while and then they can release the enzyme again to continue working. Now, the competitive is the one that competes with the substrates for the active site, which means they have almost a similar structure as a substrate, and it's about who has more. So it's a competition about the concentration. If you have more competitive, it will stop. If you have more substrate, then you will continue. So again, the competitive relies on what we say the concentration. So in another sense, if I put it here, if you have let's erase here. Let's say you have the inhibitor and you have the substrate, right? These two are competing for the active site of the enzyme. So if you, by any chance, have more substrate, then this will lead to the active site being bound by the inhibitor, and this is going to do what? Stop the reaction. However, let's do the other way. If we have here more substrate than the inhibitor, then the reaction will go in this direction. And of course, let's put it this way, the enzyme will be bound to the substrate and you will continue with the reaction. So what are we trying to say? Whoever has the more will win. So it's a competition between these two. It's a competition between the amount they have, and their amount is what influenced the reaction. So example of a competitive would be the sulfanilamide sulfur drug, which kills the bacteria during infection. So this is an inhibitor that stops the bacteria. Now, what exactly does it do? Now, we know that the bacteria wants folic acid, okay? That's like a food for the bacteria. And it's produced by the enzyme that acts on PABA. So, for example, the PABA here, right, it's broken down by the enzyme to make folic acid. And that folic acid is eaten by the bacteria so it can live. Now, what would the sulfonamide do? Now, the sulfonamide will be another type of, it will be an inhibitor, so I'm going to put here just a sulfa, right? It's a sulfa. So we have this and these two. They both are competing for this enzyme. If you have a lot more of PABA, then great, you make more food and the bacteria lives. But if you have more of the sulfa, which is the inhibitor, so they stop the enzyme, and that means no folic acid is produced. So what happens to the bacteria? It dies. So the whole point of the sulfur drugs is to compete with PABA to stop the enzyme from producing folic acid. And this way, the bacteria will die, and the human doesn't have the infection because all the bacteria have been dead. The good news with this is that PABA usually is not used by human cells. So we don't worry about the sulfur stopping the PABA at all. So here, very clear, just to make it more simple, we have the sulfur drug. And the sulfur drug is who? Is the inhibitor. Right? And we have PABA here, which is the substrate. Now, what do they do? Doing? They are competing for the active site of the enzyme. They're both competing. And whoever gets wins. So if you do have more of, let's say, of the sulfur drug, then the active site will not produce the folic acid. There's no production and the bacteria die. However, if you have more PABA, that means the active site will increase the production of folic acid and the bacteria will live. So highlighting here, if the green direction, you will go to stop the process and the bacteria die. The blue direction, which is more of PABA concentration, will cause to increase folic acid and bacteria live. So this is an example of competitive. Now, just to be clear, the sulfur drug, the inhibitor, will kind of have the same structure or similar. I would say the word similar structure, not exactly the same, but very similar their structure. And it depends on their concentration to see who's going to win. So it's really nothing to do with the enzyme. It has to do with the concentration for the both. Now, with that, then we can go for the second type of inhibition, which is the non-competitive. Now, this is not influenced by the concentration of the substrate. You don't care. You can have millions of substrate. It doesn't matter. Why? Because this non-competitive is not going to touch the active site and make it worse. It's irreversible. Once you attach, it's forever attached to the enzyme, and that means the enzyme will never work again. So where does the non-competitive inhibitor hits? It hits on the allostatic section. That means the allostatic site of the enzyme. So examples of non-competitive inhibitors is cyanide, which basically combines with iron. And what does it do? It stops the cytochrome oxidase, which is part, for example, of the enzyme reactions of cell respirations in our body and the photosynthesis of plants. We also have heavy metals such as silver and mercury. These guys, they can also stop the SH groups. What is the SH groups? That's the disulfide bonds, and that can stop production of protein. So reversible or not, allostatic reactions can activate enzymes too. So it's not always necessarily a problem. But when it comes to the non-competitive inhibitors, it always stops the process. Now, the allostatic site, you can also put a vitamin there or a mineral. so it can continue the enzyme. But if you attach a non-competitive to the allosteric site, that is over. Actually, let's show here in the picture. It does show here. So here is the active site, as you can see. This is the allosteric site, and here's the active site, and you have the substrate. So the substrate will attach to the active site and make a product. However, if you put an inhibitor right here at the allostatic site, look what happened. Look how the active site looks a shape. Look how the shape has changed. And if that change has shaped, then most likely nothing will happen to the substrate. You will not make any product. It will be stuck there, but nothing happens. This is what allostatic looks like. So if I had shown here, let's say this is an enzyme right here. And let's say, let me use here and take a little bit space here. And let's say that this. So right here, this is the allostatic site. And this is where the non-competitive inhibitor, this is where it goes and attach. Now, if it does attach, then look what happens if they do attach. It happens that it changes the active site. You see how it went from a nice smooth square to having these little zigzags. That was just because you did attach the inhibitor to the allosteric site. But again, this allosteric site was not only meant for the inhibitor. See, that allostatic site I'm going to put here is also meant for things like vitamin, minerals, and these guys can actually increase the reaction. So don't think the allostatic site is just to stop. The allostatic can also be to increase any kind of reactions. So this is again just showing that there's two receptor sites. They have the active site, which is for the substrate, and we have the allosteric site for the inhibitor or other types of molecules such as vitamin minerals to keep the reaction going. Now that we have learned the two types, let's talk about the enzyme pathway. So when you talk about cell process like respiration, photosynthesis, they have a series of pathways controlled by enzymes. So here is like we have the substrate A with the enzyme A make product B, and then product become a substrate reacting with an enzyme becomes C, and then C becomes a substrate react with the enzyme C makes a product D. Then D becomes a substrate that reacts with the enzyme D and makes product E, and then E becomes a substrate that works with the enzyme and becomes a product F. So you see a long pathway of enzymes, or should I say cascade of enzymes working and making different products. Now, of course, you can see each one is by a different enzyme, and that's because the enzymes are very much specific to each one. As you can see right here, that you have enzyme A working on A, enzyme B working on B, C on C, D on D, F eventually should be working on here, so this should be an E. Now, to continue all this is usually the start, and this is usually the end product. And the ones in the middle right here, we usually call these guys as intermediate product. So you have to know this because these kind of words can come out in IB exams. So we have the end product, which is F, the start substrate, which is A, and the intermediate product, which is between B to E. This is where we get to the point where we talk about endpoint inhibition. So usually it's the first step that is often controlled by the end product. As you can see, the product F right here, it goes all the way back, all the way, and stops the enzyme A. If you stop the enzyme A, none of this in the middle, none. All of this is completely stopped. Now, this only happens when you have such a large amount of F, then you say, hey, I already produced so much F, I don't need this enzyme, this process to continue. So I go all the way back, and through negative inhibition, I basically tell the enzyme, hey, stop, no more of it. Now, what happens if you don't do that? What if you do it beforehand, like in some other enzymes? Let's just say then instead of the product F here, it actually comes and stop this enzyme. That is not an issue because you can pretend none of this is here, right? But you will continue this process, so you're going to start increasing the amount of substrates on D. And when you have so much process of D, what happens? A D will collide with a D and it will form an E. The only difference is going to take a little bit longer without the enzyme, but it's still going to make E. The only reason why you put an enzyme is to speed it up. But without the enzyme, you still can react D with D and make an E. So if you really want to stop the whole process, you really have to stop right here at the beginning of all of it. So the end product controlling their own rate of productions. So there's no building up of the intermediates. Like I told you, none of the intermediates should be building up. They should all be decreasing. So it's on the allostatic side of the EA, the enzyme A. That's where the product F will bind to and stop it. So that means when you have a high amount of the F, it will inhibit the entire enzyme, and that means lower the whole process. But it's very important that when you think about the enzyme A, it is not attaching here on the active side. It's attaching on the allosteric side. So who comes on this active side? It is definitely the substrate A, just to make it clear. So that's what it means by the endpoint inhibition. So is the negative feedback reversible? Yes, it is. Now, the non-competitive inhibition, it is reversible. It depends on the situation, but most of the times they are not. What does the inhibitor concentration do? It just slows down the enzyme by changing its enzyme conformation. Now, if you get rid of it, you can go back to the active form, the active site back to its shape. So here is an example. You have the bacteria E. coli, which produces amino acid isoleucine from threonine in 5 steps of process. If isoleucine is added to the medium, the first enzyme is inhibited and no more isoleucine produced by the bacteria. It will go back to the producing once isoleucine is used up. So here is the process. This is one of the requirements IP is that you know this chemical reaction, where you have the initial reactant, which is threonine, and the final product, which is isoleucine, and in between them, you have enzyme 1, 2, 3, 4, and 5, and you have the intermediates A, B, C, and D as the intermediate products. Now, if you have too much of isoleucine, the isoleucine will go all the way to the first enzyme and stop the whole process because you already have so much isoleucine, so you stop all of this. But once you don't have enough isoleucine, what do? You reverse the whole thing. If the isoleucine is low, then you allow the enzyme 1 to go back to its normal and continue the process so it can produce isoleucine again. Now, calculating the enzyme reactions. When we talk about enzyme reaction calculation, we are looking about the rate. That means how fast can the enzyme reaction can be done. So once you calculate, usually this is plotted on a graph, and from there, according to the time, you will figure out how fast it is. Now, the time taken can be measured according to many ways. One of them is the amount of product that is produced or the amount of substrate that is consumed. These are 2 ways, and it's very important that you know this. The amount of product produced or the amount of substrate consumed. These are 2 ways that you can calculate an enzyme reaction. Now, reaction rate is the inverse of time taken, meaning that the reaction rate is higher when it's less time taken and vice versa. So here is the formula. The rate of reaction is basically over this time, which is 1 over the time taken. So it's always going to be 1 over the time. And this is how you figure out. So you can find the time it took for a product to make, you put it up here. Or you can find the time it took the substrate to consume, and you put it here. So let's say if the time to make a product took 2 minutes, you have to put in seconds. So that would be 2 times 60, which is 120. So it would be 1 over 120, and you will find the rate of the reaction. Or you can say the time that it took the substrates to be completely consumed was 5 minutes. So that will be 5 times 60, and then you have 300 here. Then you will put the 300, and you will find the rate of reaction. As long as you're finding one of the 2, the amount of product formed or the amount of substrate consumed. And once you find the time for one of these 2, you plug into the equation, and you will find the rate of reaction. Now, calculating the enzyme reactions here, we can look at the pH, the temperature, or the substrate concentration, right? We can always find here the rate of reaction. Here the pH, we know that pH has an optimal as well as the temperature, and the substrate usually has a plateau right here. Now, these right here is basically considered as the optimum that an enzyme will work, right? Now anything below, it will slow it down. This will also slow down, this will denature, and of course here it speeds up, but then it plateaus because that's the max amount of enzymes. If you want to calculate an enzyme reaction, we can always find at the process right here how many products have been used, or we usually can have a graph where we talk about the substrate here, and right here the products, how much product is being formed, or the enzyme activity. From this process we can figure out at here how much product is being produced after how many minutes. So I can also have here substrate over time, and then we can see the substrate if it goes down or up, whatever it is, and we can pick here the amount of substrate and the time, and from there we can put into 1 over whatever the time is and find the rate of the reaction. So looking at the enzyme graphs, we have to talk about the different types of enzymes. The one when you have the enzymes, when you have the competitive, or the non-competitive. Here we have the substrate concentration over the rate of reaction. Now this is considered as the normal graph. This graph will always be the same. Every time there is no enzyme, there's no inhibitor, that means this is what it's going to look like, a normal graph of a substrate against the rate of reaction. And what is the Vmax? That would be the amount of enzymes there is. So if there's 100 enzymes, that's the maximum will be active. No matter if you get 500, it will keep going plateau because that's the amount of enzymes there is. Then you have here the competitive inhibitor. Now competitive inhibitor, that means they're both the substrate as well as the competitive inhibitor. They're both competing for the active site. So you can see there's a little bit of a drop, but not necessarily a big drop of the reaction. But that means the competitive inhibitor is starting to slow down the reaction. And then right here on the non-competitive inhibitor, because it hits on the allostatic site, it's not competing for the active site, you can see that it slows down even faster the process. That's why the graph is lower. So you have to know these three together. So usually this graph right here, if you know it by heart, then you'll be able to answer any questions related to this topic. The green will be the one without enzyme, the blue will be the competitive, and the red will be the non-competitive. You will always find this in any kind of test. So remember, competitive will always, both inhibitors here will always be below the normal one. However, the competitive one, because it's not competing, it slows down even faster compared to the competitive, which is still competing with the other substrate. So then we come to the rational drug design. Now, malaria is a disease caused by a parasitic protozoa, Plasmodium. Now, to live, it needs human or mosquito as a host. They also need enzymes to mature and develop on the host, thus creating inhibitors for these enzymes, as an anti-malarial drug can be stopping the disease. So again, we want to create an inhibitor that will stop malaria. So it has to be an inhibitor that will stop this parasitic organism. So that means the genes and the proteins of the parasite was identified, and this way we figured out which enzymes we need to break it down. So by using the bioinformatic database, that means by figuring out what is the genes and by figuring out what are the proteins made by this parasite, right? These two, the genes and the proteins that the parasite make, then we can figure out how their enzymes function. And this way we can figure out how to create an inhibitor that will stop that enzyme. And by stopping it, we eventually have a new way of killing the enzyme. And that's a way of calling the rational drug design. It's just finding the right inhibitor that will have a stronger affinity to bind to the enzyme and stopping the whole process. So here is just showing the rational drug. We have different types of proteins produced at different stages of the mosquito, right? So here we have the mosquito at the gamete stage. This is what the proteins it produce. And then at this stage, these are the proteins it produce. And then right about this stage, these are the proteins they produced. So we have different types of proteins at different stages of the mosquito, right? So what we do, you can produce inhibitors for this stage, inhibitors for this stage, and inhibitors for this stage. That's why the doctor will give you a certain type of medicine depending on what stage you are. And when we do that, these inhibitors will stop these proteins right here. And when they stop these proteins, then the mosquito will not be able to live. That's simply what it means. But all of this can only be done if first we figure out all the genes and all the proteins, that means including the enzyme of the mosquito, how they function. Once we figure out how the enzyme functions, then we can create an inhibitor artificially and give this inhibitor as a drug to kill the mosquito. But remember, this inhibitor will affect the enzyme of the mosquito and not the enzymes of the human body. Because you can see here they have different types of proteins. So these enzymes inhibitors will be affecting here, but will not affect our proteins production. And that's pretty much it for the metabolism topic 8.1. Here's a study guide. All these questions here have been answered in this video. If you're able to answer these questions, then you have a strong base for 8.1. Thank you.